There we go. Welcome, everyone. Thanks for joining us today. We'll give everyone a minute to get settled here. All right, for the sake of time, I'll kick us off for today. So welcome, everyone. Thanks for joining us for today's Cancer Immunotherapy 101 webinar. I'm really excited to have a fantastic crowd here today. My name is Julie Jonkins. I'm the Training and Research Manager at BioCanRx. And for those of you who might not be familiar with BioCanRx, we're a federally funded national network of researchers working together to accelerate the development of leading-edge immuno-oncology therapies for the benefit of Canadian cancer patients. We invest in translating world-class technologies from the research lab into clinical trials, and we also train and develop the talent needed for a thriving Canadian health biotechnology sector. And our vision is to turn all cancer into curable disease. I'd like to acknowledge the land that BioCanRx is located on. We're based out of Ottawa and Ontario, and we're situated on the traditional unceded territory of the Algonquin and Anishinaabe people. We acknowledge and respect our traditional hosts and thank them for allowing us to operate on this land. Now, I'm very pleased to introduce today's speaker, Meghan Kates. Meghan is a PhD candidate in the Department of Immunology at the University of Toronto, supervised by Dr. Samuel Sabil and Dr. Pamela Ohashi. Her research focuses on understanding how changing the metabolism and health of patient products can improve T-cell adoptive cell therapy for patients. Meghan is also very interested in making science accessible, and she's actually been delivering this talk to our patient scholars in the BioCanRx Learning Institute for several years now, and she does such a fantastic job at this that we thought, why not make this session available to more people and get more people involved and have more awareness of cancer immunotherapy. So we hope that this is a valuable learning opportunity for you all today. A few quick housekeeping notes before I turn the floor over to Meghan. Meghan, I think, will be pausing throughout her presentation to take questions from the audience. So please use those times through the Q&A or either raise your hand and we can unmute you to ask your questions. And also, we'll be recording the session and we'll be posting it to our website afterwards. So it's a resource that you can return back to. So without further ado, I'll turn the floor over to Meghan. Thanks, Meghan.
Thanks, Julie. Let me get my presentation shared here. Okay, hopefully that looks okay. Yes, okay. So good afternoon or wherever you're from. I hope you're having a good day. I'm really pleased to welcome everyone to this Immunotherapy 101 session. So Julie's really already done the introduction for me, but I had a couple of photos here of myself in January presenting at a conference and where I'm working at the Princess Margaret Hospital. So this is part of the University Health Network in Toronto, and I study cancer immunotherapy in the context of melanoma. So the agenda for today, we'll talk about an immunology primer to really get everybody up to the same speed on immunology and the background that we need. We'll talk about immune escape in the context of cancer and different types of immunotherapy. And finally, we will end with hot topics in science and immunotherapy. So this talk, as Julie mentioned, is one that I've given to the patient scholars for the BioCanRx Learning Institutes. So adapting it a little bit today, and I understand that everybody's probably coming in with different knowledge on immunology, as well as on a broad range of these topics. So I am going to start from a very foundational level of immunology, and we'll work up together, so hopefully we all can end with a very similar knowledge base. With that in mind, hopefully it's not too tedious for those who know some of these concepts, and hopefully we can get everybody up to speed the same way. So to start out, I really just wanted to ask a question here so I know where your knowledge starts. So what do you know about the immune system? You can actually put this into the Q&A, and I'll try and take a look at that as well. So any types of cells, anything that you might know that the immune system does, anything at all, or how it works in the context of cancer. I don't know if we're getting any responses. I don't know if everyone's being very shy or I cannot see it. It's also possible too. I don't know if I have the power to unmute, but if you can go forward. Try.
I was just gonna say the chat is disabled, so we can't use it.
Yes, I think it's through the Q&A function that we're going to try. Is that one also disabled? Oh, here we are. Got a few in
Let me
here.
check. Okay, perfect.
Thank
you.
Thank you. Alicia, I would assume that it is our body's natural ability to fight infections and other bacteria. Absolutely. An issue with the audio. I don't know if you guys can react, if you can get a thumbs up or a thumbs down if you can or cannot hear the audio.
I think that one was resolved. We're all good there, Megan.
Perfect. Okay. Okay, so definitely we are starting with a fundamental understanding that the immune system is our body's way to fight infection and other bacteria. So that's definitely a great place to start. I think everyone else is just being a little bit shy. But just starting from that place, what is the immune system? So this helps to protect your body from foreign invaders, as well as to help maintain homeostasis. So I think this audience, you probably know pathogens are organisms that can cause disease to the host. This can include a number of things from fungi, bacteria, parasites, and viruses. And your immune system is going to help protect you from all of that, as well as to keep things going in the way that they should, so maintaining homeostasis. There are two broad categories of the immune system that we'll talk about today, both the innate and the adaptive immune system. And if we just take those words as they are, I think that they're very informative. Innate means that it's already present, so it's already there to respond to threats. The adaptive immune system, on the other hand, is going to be able to adapt and change as it still has threats come up. So the innate immune system, therefore, are already there. They're the first responders, and it means that they're a quick response to any danger that you might face. But the innate immune system, the trade-off is that it can be a little bit more short-lived. The innate immune system also isn't specific to a particular pathogen like a bacteria. What it will recognize is a broad category of danger, such as maybe bacteria in general, and try to stop that you encounter it. And what the innate immune system can do is it's short-lived, but it can then recruit the adaptive immune system in order to be able to continue the fight. The innate immune system includes both barriers to pathogen entry, as well as cells to eliminate pathogens, and we'll talk about both of those in the upcoming slides. So what the innate immune system does is it prevents pathogens or dangers from spreading in the body. And if it's overcome, it can also hand things over to the adaptive immune system. These are second responders to threats, but they take a little bit of time. They're a little bit slower in order to get recruited. The interesting thing about the adaptive immune system that we'll talk a little bit more later in the lecture is that it has memory, so it has the ability to prevent reinfection from something you've seen before. And it is very, very specifically targeted using specialized cells in order to have that targeting. And we will talk extensively about the cells in the adaptive immune system. Overall, the adaptive immune system will eliminate threats that have spread and remember previous threats in order to react faster upon reinfection. So the innate immune system, I mentioned it's made up of both barriers to pathogen entry, as well as cells to eliminate pathogens. Let me grab my mouse over here. So some barriers that you might have already thought of are your skin. So you can prevent pathogens from entering just by having a layer covering your body. So you are not going to have anything crossing through unless you have something like a wound that would allow something like bacteria to cross over. Slightly gross, but mucus also is a layer to prevent pathogens from entering the body by trapping them. This is both in your respiratory system as well as in your gastrointestinal system and others. Your eyes as well have tears to flush out pathogens, as well as enzymes called lysozymes that can help break down those pathogens. And finally, stomach acid can prevent pathogen entry through the gastrointestinal system by breaking down any of these pathogens so that they cannot enter and infect the host. So these are just to keep things out. We don't want any pathogens to even come into the body. But if they are there, then what do you do? So there's a number of cells in the innate adaptive immune system. And I have a few that are listed on this slide, some of which I'll cover just as a way to make this a little bit more complete, but we don't often talk about them in the context of cancer. So this is not really where the focus will be. So that's these first ones here. Ones like neutrophils, that someone made the analogy of Spider-Man, which I thought was excellent. This is because they actually cast out webs to trap pathogens, and they can also eat small things and break them down. Basophils are immune surveillance cells, and they're often involved in both allergy and asthma. And eosinophils have a role in killing parasites. Again, these are ones that really we don't tend to think about too much in the context of cancer and probably won't come up at the conference. So we can leave those ones as is. Ones that definitely will come up a little bit more are things like macrophages. And the name itself actually really means big eaters. So these cells are actually capable of taking in and breaking down or eating things like pathogens to get them out of the body and out of the way. Natural killer cells, also known as NK cells, we can call these generic killers. So they will recognize broad categories of threats and be able to eliminate them. These broad categories of threats can include things like virally infected cells. They understand, hey, this is a virus, this is not supposed to be here, and can eliminate those cells. They can also recognize certain tags that are produced by the immune system, which we'll talk about a little bit later. But when it finds something that's tagged, it can then eliminate that tagged cell. And finally, dendritic cells. These ones we call DCs, and you can think of them kind of like educators. These cells, you can see they have long tentacle-like appendages or dendrites, and what these are doing is surveilling the environment and looking around at what's there and taking in little bits from the environment. What it's going to do with that is actually show it to the adaptive immune system and be like, hey, this is what's going on in the body. Do you think this is a problem? So that's one way that the innate immune system can actually have crosstalk and interaction with the adaptive immune system by using DCs to actually show what's going on in the body. I guess I can pause here for a moment and see if there's any quick questions about the innate immune system before we move on to talk extensively about the adaptive immune system. So you can either put that into the Q&A if you'd like to type it out, or raise your hand if you do have a question so far. Okay, clear as mud. Okay, so I will have another pause point after talking about the adaptive immune system, so you can also hold any questions until then. The adaptive immune system, on the other hand, I used this analogy for the patient seminar, and I thought it was very useful as well. You can think a lot of the interactions in the adaptive immune system function like a lock and key, where you have to have a very specific key to interact with the lock in order for anything to happen. If there are people here, I know many of you work in science, you might know receptor-ligand interactions. Very similar concept here in the adaptive immune system, where you get that receptor ligand or lock and key interaction. You need to have that very specific recognition in order for anything to occur. So there are two cells that make up the adaptive immune system. The first one here, which I'm sure you have heard of in the context of cancer, are T cells. We can call these ones serial killers because they are capable of identifying a very specific target and killing many of that target. They have a molecule called the T cell receptor, or TCR, and that's that receptor or puzzle piece lock and key interaction that they can have. This allows them to recognize very specific threats. You can think of this cell over here, maybe it's a cancer cell, and when that interaction happens, that's when the T cell is able to kill that cell. So these are really the warriors of the adaptive immune system that can go out and find problems and eliminate them. So how does this interaction happen at a little bit more of a granular level? There are several signals that T cells need in order to activate to be able to kill their targets. That first interaction happens through the T cell receptor, or TCR. And what it's doing is looking for its very, very specific thing that it recognizes. I also use the example of a puzzle piece interaction, puzzle piece, lock and key, receptor ligand, whatever is easiest for you to think of. So the puzzle piece or the ligand that it's looking for is these molecules over here. So what's happening in this purple cell over here, you have this blob, it's a type of protein, for example, and your cells are going to process these proteins and show it to the immune system to ask if there are problems here. Is there something inside the cell that's actually a problem? So it can actually process these proteins into very small chunks called antigens that it can then show to the T cell in the context of this molecule over here, which is an MHC or HLA. And don't worry too much about what it stands for, but what you can think of is this MHC or HLA is actually expressed by all of the cells in your body, and it can show T cells what's going on inside to ask if there's a problem. Most of the time, no problem, and the T cell is not going to interact and activate. Other signals can really help to boost this activation. Things like co-stimulation, which are several other different receptor ligand interactions. So lots of other different puzzle pieces, as well as cytokines, which are instructions for the T cells on how to perform their function. We can look a little bit more granularly at this TCR and MHC interaction. So I liked this diagram over here. So they've just kind of reversed the image here. So on top here is the T cell. This green bit here is the T cell receptor or TCR. And the bottom cell over here is showing a peptide in the context of an MHC. So it's this very, very specific interaction and recognition that allows for a T cell to activate. So this very small region of peptide that was processed from a protein is what's recognized by the T cell and the TCR to activate. But how do we actually have the ability to recognize all of these different things? So to be able to recognize any possible peptide or antigen, T cells will actually, during development, undergo random recombination in this very specific recognition region, and allows them to generate a random sequence that can recognize pretty much any possible type of peptide that you might show it. So if you do the math, that's actually 10 to the 14 to 10 to the 19 possible sequences. So it's an unfathomably large number of sequences that T cells can recognize. And that can allow them to recognize anything that might be a threat to your body. And this is very specific. Each T cell is really going to recognize one antigen. So you're going to have lots and lots and lots of different T cells that each individually recognize a very specific thing. But when it encounters its target, that's when they can start to identify that threat, grow and expand their army so that they can go and deal with it. So that's one of the reasons it can take a little bit of time for your adaptive immune cell to function. They have the arsenal, but they need to really build it in order to be able to have an effective response. So that was a lot of information about T cells. I promise you there's only one other cell in the adaptive immune system, and these ones are B cells. So B cells, very similarly to T cells, have a receptor that allows them to recognize certain things. These are usually things that are on the outside of a cell, or maybe like a toxin or something in the body that it recognizes. And these little Y-shaped molecules you might recognize if you're working in science as antibodies. But these can be called B cell receptors when they're actually on the surface. When a B cell is activated, it will actually secrete these antibodies into your system so that they can recognize whatever it is that they recognize anywhere in the body. Again, if you work in science, you might recognize antibodies that we use in order to recognize very specific proteins or different things. But what actually is the role of an antibody in the body in the context of your immune system? So antibodies can perform a number of different roles. One of them is for neutralization. So if you look at this image over here, you can see these little red circles are a type of toxin. Antibodies could recognize a toxin, surround that toxin, and take it out of your system so that you are not going to have that toxic effects. they can then be cleared by things like macrophages that are just going to break down and eat it, so it is not a problem for the rest of your body. Another role of antibodies, instead of neutralizing something like a toxin, they can also coat the surface of something that's harmful, like a bacteria, through a process called opsonization. Really just means coating the bacteria. And then these antibodies can actually get recognized by your innate immune system as a tag. When I was talking about the innate immune system, I mentioned that NK cells and other cells can recognize tags. Antibodies are these tags generated by your adaptive immune system to flag this to the rest of your immune system as a problem. Like, hey, this is not supposed to be here. You should get rid of it. So things like NK cells or macrophages can come along to destroy or eat or phagocytose anything that is a problem here. You can also activate other aspects of immunity and signaling pathways like complement, but we won't talk too much about that today. So overall, these antibodies can be used in the immune system to neutralize or remove threats from the body. Another important feature of the adaptive immune system that I mentioned is the ability to form memory. So once you're exposed to an initial infection, you might get a relatively slow, a bit of a lower response to this infection, and you'll have a primary response and hopefully to clear that infection. After you clear the infection, most of the T cells or B cells will die, but some of them will persist as memory cells, and they can respond faster to reinfection. So that's where you get here a secondary exposure, and you can have a very fast and very high response to that infection in order to be able to clear it even faster. This can even give you lifelong immunity in some cases. And we definitely take advantage of that when you think about vaccination. Vaccines are small, inactivated components of a virus that are used to stimulate an immune response. So you can create those memory cells that you need for faster response, but without getting symptoms of the disease in the first place. If you then encounter that virus again, your cells will remember it and respond very quickly to reinfection. You might not even get symptoms of that infection at all. So this sounds awesome. There's lots of incredible things that the immune system can be doing, but we have to be careful because immune regulation is exceptionally important, and your immune system does have the potential to cause harm. So in order to prevent harm, the immune system has to be regulated so it can do two key things. The first one is to differentiate between things that are self, so part of you, these are good, we don't want to attack these, and then things that are non-self, like that bacteria, for example, so that we can attack and get rid of these. The other thing the immune system needs to do is be able to hit the brakes when it's no longer needed, and not just keep running and running, because that's going to take energy and lead to problems that you don't need. So if we think about how the immune system can go wrong, and what are some of the outcomes of this, one of them that might come to mind are autoimmune diseases. So these can be where your immune system is actually attacking itself. So that first bit of regulation that it's not properly doing in particular cases. So you might get recognition of small pieces of different parts of the body, leading to particular autoimmune diseases, such as multiple sclerosis, if the nervous system is targeted, type 1 diabetes in the endocrine system, ulcerative colitis or Crohn's disease in the gastrointestinal system, rheumatoid arthritis, psoriasis, etc. So that's one very important piece of regulation for the immune system. The other problem with the immune system is when you can't hit the brakes or be able to tell your immune system, we don't really need to do that. So one case is allergy. So if you think about allergens, these are things that are external to your body, like peanuts, like dust, like pet dander, all of these things that most people don't react to. Because if we're shown these things in a circumstance where your immune system is saying it's not a problem, that's what it's supposed to do. It's like, okay, I'm seeing these things that are external to me, but don't worry about it. It's not a problem. And allergies will happen when your immune system will react to it, even though it's not supposed to be a problem. So you can get the overreaction of the immune system in that way. Another very severe overreaction of the immune system as well is cytokine release syndrome, which some people think about often in the context of cancer therapy, but occurs in other cases as well. This is when the immune system reacts too aggressively to an infection or treatment, leading to the release of an abundance of chemicals like cytokines that really overactivate the immune system and other systems in the body and can lead to quite a number of issues. So this can happen in CAR T-cell cancer treatment. So that's a therapy we'll talk about a little bit more today. It's happened definitely in the context of COVID-19 infection and even in flu infection in some individuals. Symptoms of cytokine release syndrome, or CRS, can include fever, nausea, vomiting, muscle pain, low blood pressure, swelling, shortness of breath, and actually can lead to death in some extreme circumstances. So we have covered a lot of information so far just on this summary of the immune system. So I just want to review here. We've talked about two arms of the immune system, the innate immune system, which are the first responders to threats, non-specific, but respond to broad categories of threats. And these are made up of both barriers to pathogen entry, as well as cells that deal with pathogens once they have made it inside. We've really highlighted a few cells that will come up again and are important. So ones like macrophages, they eat big things, NK or natural killer cells, which are really responsive to those tags like antibodies, and dendritic cells that educate the adaptive immune system on what's going on in the body. So these ones cross over to interact with the adaptive immune system, which are the second responders to threats, are highly specific to a very small antigen or region of recognition, and they have the ability to develop memory. These are made up of both T cells and B cells. And don't forget that immune regulation is very important. So because we've covered so much information, I do want to pause briefly here to ask if there's any questions so far before we really start to apply this to the context of cancer. So again, please feel free to put your question into the Q&A or to raise your hand and you can be unmuted. Yes, you can. Sorry if I'm saying that wrong.
Thanks, it's Bakun. I'm just wondering for the antibody receptors, how that's related to antibody conjugates, or is that specific to when it becomes like an ADC for cancer?
So antibodies themselves are essentially just those Y-shaped molecules that you'll see, used in the body, also used extensively for research. I'm pretty sure what you're talking about, the antibody drug conjugates, the ADCs, are usually when you actually have the antibodies and then you complex them to a particular drug. So what this does is allows for that targeting of that Y-shaped region of the antibody, so it can bring it very specifically to something that it's going to recognize, but then you have that payload attached so that the drug can then be delivered. I'm pretty sure that's the context of what you're talking about.
Thank you.
Thank you. Any further questions? Well, this might be review to some of you, or maybe I've overwhelmed you entirely, but hopefully we can all get to the same track in the tumor immunotherapy region as well. I don't know if I can't see any hands on other pages, but feel free also to drop questions in the chat. You can even do it when I'm talking, and then I can answer them at a break if that works. But if there's no questions at this point, we can move on to how the immune system is involved in cancer. So we've talked a lot about the immune system, and it seems pretty good at what it's doing. It can find things that are a problem. So why does cancer develop? This is clearly a problem. Like, what is it that the immune system is missing in this regard? So there's been a leading hypothesis which has been discussed for a long time called the tumor immune escape hypothesis. And of course, this is still an area of ongoing research, and a lot is being discovered about it all the time. But what it's been broken down to is essentially 3 general phases of tumor immune interaction. So the first phase is an elimination phase. So this is where the immune system does respond to react to cancer and kill it in its very early stages when you start to get aberrant or malignant cells. So this is an elimination phase. If there are some cells that then survive that selection or maybe are selected for in that process, you can start to reach an equilibrium with the immune system where there's very few of those malignant cells that are present, but they can continue to mutate and divide. And the immune system, although it's doing its job, maybe is starting to lessen as it doesn't have its target. And this can eventually lead to this escape of immune system control, often by acquiring new mutations or by suppressing the immune system itself. So how can this actually happen? And there's a few different mechanisms that lead to this response. So cancer can both reprogram as well as evade the immune system in order to escape and form a fully functional tumor. So one way this can happen is through what we call loss of antigenicity. So essentially loss of ability to generate an antigen. Remember, antigens are things that your T cell receptors are recognizing. Normally you have that puzzle piece, you have that ligand receptor interaction, and that allows you to eliminate cells that have that antigen. But that can be a selective pressure, so that cells that can actually decrease the amount of antigen or downregulate it, those ones can actually escape the immune system because they no longer have an antigen that your T cell is going to recognize. So there's nothing to target at that point. So that's one way that the tumor can escape recognition by the immune system. Another way is by loss of immunogenicity, or ability to generate an immune response. So cancer can actually co-opt a lot of pathways that exist in the body that are part of regulation. So T cells, again, need to be able to hit the brakes so that they're not too functional in the environment and leading to damage. So one way that this can happen is by having receptors that check in with the body to see if they're still needed. So this includes this receptor here, PD-1, or programmed death 1. It's a little bit of a misnomer, it doesn't always lead to death. But what it does is it is a checkpoint for T cells to ask if they're still needed or if they should really ease off and hit the brakes. And what cancer can do is actually express the ligand for this receptor, specifically PD-L1, and then that interaction can happen, leading to what we call T cell exhaustion, or a state where they're too tired to perform their function, and essentially they're no longer functional and no longer able to kill. So that will stop the immune response from being generated. A third way to evade the immune system is by overall development of immunosuppressive microenvironments. So there are many immune cells that exist within most of the tumor microenvironments, and these can include things like T cells, like macrophages, and many, many more. But tumor cells can also send signals to things like your macrophages and actually convert them very slowly to the dark side, so that they're also involved in sending suppressive signals to T cells so that they become no longer functional or no longer able to perform their function. Any quick questions about tumor immune escape before we talk about therapies? Okay, very shy group today.
I think there's actually one question in the Q&A. Is that a new one?
Perhaps it's not loading for me.
Does the immune system react differently in early stage versus metastasized cancer?
Oh, awesome. Thank you for pointing that out, Julie. Yes, the immune system can definitely respond differently in the early stages versus metastasized. So when you have a metastasis, you have often sort of already lost part of the fight because you have developed a local tumor and then it is now spread to a distant area. you've already had probably an immune response, and the immune system has probably been fighting it at this point. So it might have already performed some of these things, like lost the antigens. It might have had this suppressive microenvironment. So it might have already suppressed the immune system in that local area, but as well as systemically. So a metastasis can often not have as much of an immune response. I do think that there's still a lot of ongoing research into discovering the difference in immune response between a primary tumor and a metastasis, but that's my general understanding. Thank you for the question. Okay. So let's move on to talking a little bit more about therapy. Sorry, I'm just looking for my window here. There we go. So we'll talk about several categories of immunotherapy. So the first one here are monoclonal antibodies, and a very important subcategory of that group, which are immune checkpoint inhibitors. We'll talk about several different types of cellular therapies, and just a small little aside about oncolytic viruses. So starting with monoclonal antibodies. So as hopefully you remember, monoclonal antibodies, or antibodies in general, are produced by B cells naturally in the immune system. But researchers can also co-opt this process and produce antibodies against things that we specifically want them to react to, or we want more of those antibodies. So what we want to do is be able to target something that is expressed highly on a tumor cell, but not really on a normal cell, or at least to a very low degree, so you can get interaction specifically with that tumor cell. One example of this is the HER2 receptor that's found on a lot of breast cancers as well as some ovarian cancers. And this is actually a growth factor receptor that exists normally in breast tissue, but is highly expressed in some breast tumor cells. So if we actually block this receptor using an antibody, because this is a growth signaling receptor, if you block that signal, you block the ability of them to grow. And because it's more expressed in the tumor as opposed to in the normal cell, this should have mostly selective pressure towards those tumor cells and death of those tumor cells. So how do monoclonal antibodies actually lead to the death of the tumor? So one way this can happen was what I had already mentioned. You can either block the receptor or the ligand. In this case with the HER2, because it's a growth receptor, you block that growth signal, you don't have growth, and eventually can lead to death of the target cell. But there are other ways to use monoclonal antibodies in order to be able to kill tumor cells. You can have antibodies that recognize something that's not a growth factor receptor on a tumor cell, but what it can do is actually provide a tag. So remember, those natural killer or NK cells are actually capable of recognizing these antibody tag cells, and it knows, hey, that's a problem, I need to eliminate it. So by putting that tag onto the tumor cell, you can cause the death of the tumor cell. Very similarly, we can use that antibody tagged tumor cell to be eliminated by macrophages, or those big eaters, instead. So just different ways of leading to that cell death. We can also lead to other pathways like complement, but again, that's a little beyond the scope of this talk today. So this is really, so far, all about how the innate immune system interacts with these antibody tags in order to lead to tumor cell death. And just a short definition here that you might already know. Monoclonal antibodies are usually lab-produced antibodies that bind to specific targets in the body to kill or to neutralize them. In the context of cancer, of course, we want to kill or neutralize those tumor cells. But there's also a type of monoclonal antibody therapy that is very important for the adaptive immune system. So this is immune checkpoint inhibitors, and they function in the context of T-cell exhaustion, which I had briefly mentioned before. So remember, T-cells want to check in with the body to know if they really are needed to perform their function. One way they can do this, and one receptor that they use for this, is PD-1. And if they have that interaction, that lock and key puzzle piece interaction here, that's where they will decide that, you know what, I am exhausted. and that's a state where T cells are no longer functional. There's a ton of cells in the tumor microenvironment that exist and can give these sorts of signals to T cells to really make use of this normal pathway to shut down T cells in order to allow the tumor to grow. So they're co-opting that pathway. So for the case of therapy, one thing that we can do is block that interaction between the exhaustion ligand and receptor. So there's several different interactions that can occur that can lead to exhaustion. And immune checkpoint inhibitors, or what we call checkpoint blockade, are antibodies that bind to a specific immune checkpoint molecules and act to block these exhaustion signals. So for example, we can have antibodies that are targeted to block signaling to PD-1, or they can target the ligand here and block PD-L1 so that signal does not occur. There's many other different exhaustion or inhibitory signals, such as CTLA-4, that can also be blocked by antibodies. So here's just a short list of some of the antibodies that are used commonly in clinic to target these different molecules or these different checkpoints. So PD-1 can be targeted by the monoclonal antibody pembrolizumab or nivolumab. The more patient-facing or the drug name for it, either Keytruda or Opdivo. The common PD-L1 antibody is durvalumab, also known as Imfinzi, and the CTLA-4 targeting antibody is ipilimumab or Yervoy. Don't ask me why they have complicated names. That is in the realm of the pharmacists who are designing these, or the chemists. But we often see these used actually in combination with each other as well. If you ever hear someone talking about an ipi-nivo treatment, that's actually a combination of these two different checkpoint blockade molecules. So this is both a blockade of ipilimumab and nivolumab, blocking CTLA-4 and PD-1. So these are used in the context of a variety of different cancers and in different combinations. And these can actually have great success in a lot of patients. But there are definitely some patients who still do not respond or who have toxicities that are associated with these drugs. So there's still a lot of ongoing research to try and figure out which are the best combinations, as well as to develop new combinations by looking at these very complicated interactions that can lead to inhibition of T cells. So there is another one that has been more or less recently. I think I had 2023 that this was FDA approved. This is an inhibitor of this is a immune checkpoint inhibitor that inhibits this checkpoint here, which is LAG3. And I am not going to attempt to say this one. But there's definitely a ton of ongoing research into a lot of these other interactions to see if that can lead to improved therapy. So monoclonal antibodies in general have been used for a lot of different applications. So these are the monoclonal antibodies listed here. This is what they're targeting. This is the type of antibody and the indication or the cancer that they're used for. You can see across a number of years, there's been a lot of monoclonal antibodies used in the context of cancer, many of which to very successful application. So I do have scheduled at this point for a break. I don't know if we want to take this. We should have a little bit of time if people want a little bit of a breather. But Jaden or Julie or Mackenzie could maybe comment there.
Yeah, I would say we could maybe keep going just to have more time at the end for questions if people want. It'll also be recorded, so if people want to take a break and come back. I'm seeing some thumbs up as well, so that's good confirmation.
Perfect. Haven't overwhelmed anyone too much yet. There is still time. So we have talked about one category of therapy here. These are particularly the monoclonal antibodies. So we can actually jump right into the next category of therapy, which are the cell therapies. So these broadly involve the use of patient-derived T cells that are activated and expanded ex vivo and then reinfused back into the same patient. So you see here T cells taken out of the patients and then they're expanded and given back to the same patient. So it is a very labor-intensive process, that is manufactured for each individual patient. There are several different types of cellular therapies with these lots of different acronyms here: TCR-T, CAR-T, and TIL therapy, which we will go through. So talking about the first two types of therapy here, the TCR or CAR T-cell therapy. So this starts off by taking T cells from the blood of a patient. So these T cells they're just floating around there, they're minding their business, they might not be actively involved or able to recognize the cancer. So we actually have to give them that ability. We're like, okay, you are now selected, and we are going to give you the ability to recognize the cancer cell, expand you, and then give you back to the patient with that ability to have recognition. So there's a couple different ways that we can actually give T cells that recognition. One of which should be relatively familiar, this TCR or T cell receptor. So that's one type of therapy. So we can actually engineer T cells to have a very specific T cell receptor, recognizing something that we know is expressed on that patient's cancer cells. So this is a very specific thing that would be expressed in the patient, so highly in the tumor cell. And it's expressed in the context of MHC. So just to remind you where you've seen this in the presentation before, we talked about that first signal that's needed for T cell activation. We have interaction between the protein that is processed into an antigen and presented in the context of MHC or HLA and recognized by the T cell receptor, followed by other signals that lead to full activation of a T cell. So that's one thing that we can give to T cells to equip them with the ability to recognize tumors. Another thing we can do is give them a different type of receptor, or a chimeric antigen receptor, which we call CAR. So these actually use small fragments of antibodies and domains that allow for signaling. So ability to recognize and then signal for activation. So it's a whole construct that we can put into the T cells to allow for that recognition and activation. So this can usually recognize things that are on the surface of a cell. So they're just kind of recognizing different pools of things here. But both of these have T cells coming from the blood. We give them the ability to recognize the tumor by something that is overexpressed or high in that tumor. There is a third category of cellular therapy. This is TIL, or tumor infiltrating lymphocyte therapy. And I did have another diagram here in a minute to show that as well. So instead of taking T cells from the blood that we think probably don't have specificity for the tumor, what happens instead in this therapy is they actually take a fragment of a patient's tumor and then use the T cells that are in that environment and expand those ones. So it's assumed that some of those T cells, because they're there in that environment and hopefully expanded in that environment, that they actually have a specificity for things that are on the tumor. So then you can hopefully expand a pool of tumor-specific T cells and give those back to the patients along with signals to survive. So if we look at it again in this diagram, you have a fragment of tumor taken out of the patient. The T cells that reside in that environment, hopefully many of which are tumor-specific, can then be expanded and given back to the patient with ability to survive. So just sort of a different way of targeting the tumor. So a little bit of a summary of these therapies. So TCR-T or CAR-T therapy, so T cell receptor or chimeric antigen receptor therapy. So these both involve manipulating T cells in the lab by giving them a receptor that can recognize the tumor. They do recognize slightly different pools of antigens. So this, the TCR is recognizing things that come and are from inside the cell and are processed and shown. Whereas the CAR T cells can recognize anything really on the surface of the cancer cell. So TCR-T cell therapy was FDA approved in August of 2024 for a particular soft tissue tumor soft tissue sarcoma, a metastatic synovial sarcoma. And the CAR T cells in particular have really high success in blood cancers like lymphomas and leukemias. Not as much in the context of solid therapy, but that's a huge area of research as well. So there is FDA approval just in that specific sphere. So there are definitely associated costs as well as toxicities with these therapies. So because we are giving so many very active T cells to a patient, you can have just a lot of signal going on, including and up to the very scary side effect of cytokine release syndrome. But you can also have other interactions as well, some a little bit more minor. You can have things like fatigue or a rash, but you can even have up to neurological symptoms as well. So definitely ongoing research also in that regard to be able to mitigate those side effects and have the benefit without some of those scarier side effects. TIL therapy, on the other hand, or tumor-infiltrating lymphocytes, and I forgot to mention lymphocytes. This just really means T cell or B cell, but in this context, we'll talk about it in T cells. So these are tumor-infiltrating T cells. So that is what you're expanding in this case. So you're not actually manipulating these T cells in the lab. This therapy has actually shown about 50% efficacy in metastatic melanoma, even for patients who have been pretreated and have failed on other types of therapy, even immunotherapy. There was finally FDA approval for this therapy in March of 2024, particularly for a case of melanoma, I believe. I don't know if I have this written here. So the drawback with this therapy is that you're assuming that there is specificity for the tumor in these T cells, but it could be the case in some cancers that maybe there's not a big pool of them. So that's something that definitely is part of what you would be looking for. There are also therapies that are there are also toxicities that are associated with this therapy, particularly because you're giving a cytokine, the cytokine IL-2 with it, which can lead to some issues in patients. Very similarly, this can also lead to just kind of an overactivation as well. Just checking the chat here, I do see questions come in. So if exhaustion signals are blocked, will that make your immune system overwhelmed? And is there some negative effect of this? Definitely, there can be. There are side effects, unfortunately, associated with all of these therapies. So it can overwhelm your immune system, as in it can be either a little bit too active, but also the effect maybe doesn't last for that long. So hopefully you get really good response. Your T cells overwhelm the tumor and are able to clear it or put it into remission. But unfortunately, that's not the case for all patients. Either they can have toxicities associated with it, they don't respond to therapy, or they respond to therapy for a while and then they have relapse. So again, tons of ongoing research into all of these different things. FDA approval, as you can see here, hasn't actually been that long, and then Health Canada approval often a little bit delayed from that as well. But still tons of ongoing research into all of this. Okay, so a lot of information about cellular therapies. I promise there's just one very quick aside to what I think is a very interesting therapy, but isn't used really very much in clinic. And these are oncolytic viruses. So this all stemmed from this observation in 1890 by Dr. William Coley, who discovered that some patients with cancer would temporarily go into remission after viral infections. So this kind of comes back to this idea of viral replication. So viruses don't have the machinery to make copies of themselves. They actually have to get into host cells and make copies of themselves in that way. So you can see a virus here attaching to and releasing its information, its RNA, into a cell, and using the machinery that exists within that host cell in order to make more copies. So sometimes these viral particles can just be released, but often they can lead to death of the host cell. So this is what's happening also in the context of an oncolytic virus. So it's kind of what it sounds like. Lytic, meaning lysing or killing. and onco meaning cancer, so viruses that can kill cancer. So some of these can be more naturally existing, as more sensitive towards cancer cells. We can also engineer viruses in the lab to be able to target tumor cells specifically, either through molecules that they overproduce. So they can recognize the tumor cell by things they overproduce, but not normal cells which don't really have that same expression. So they get really selectively into tumor cells. You can also decrease the aggressiveness in normal cells, maybe by using machinery that's often in tumor cells, but not as much, again, in normal cells. You can also ask these viruses to express tumor-killing factors to lead to that killing of the tumor cell when it infects as well. Oncolytic viruses are capable of infecting cells and killing them directly, as you can see in this diagram here, but they also release a lot of different particles that can really inform the immune system of what's going on and maybe reinvigorate it, and lead to clearance also systemically in distant metastases. It's definitely a very interesting therapy, and there is FDA approval in some circumstances, but it's not really used very broadly in clinic, potentially due to short duration of the effect, or maybe even issues with manufacturing the virus. I'll admit this one is not my area of expertise, so while it is an interesting aside, it's probably not something that you will come across too much. But what we do see a lot, and really a growing area of research here, is combination therapies. So how can we improve patient therapies by discovering what combinations, first of all, work well together, but also will work specifically for an individual patient? Very important avenue of personalized medicine that is emerging in this day and age. So we've talked a lot about therapies, going through first monoclonal antibodies and a very important subcategory, which are immune checkpoint inhibitors. We've talked about 3 different types of cellular therapies: TCR T cells, CAR T cells, and TILs, lots of acronyms, and also just briefly about oncolytic viruses. I can pause here for a second before I go into the hot topics of cancer therapy to see if there are any more questions here. Okay, not at the moment. So we can definitely loop back to some more questions, or if you think of some in the meantime, you can put them into the Q&A. I'll just run through the last area of this talk, which is a few hot topics in science and immunotherapy. So these are things that are specific to the different plenaries at the conference. So some of this you might be more or less familiar with, but just a little bit of a review before we jump into the conference next week. So one topic that will be at the conference is about mRNA and lipid nanoparticles. So many people, especially when I was doing this lecture right around COVID time, would be like, wait a minute, I've heard of this. What was this from? And many individuals had heard this through the COVID vaccine, which was a vaccine lipid nanoparticle design. So you can see here what is covering this area is lipids. So this circle with the 2 squiggles are your lipid bilayer, which is also a component of your cells. So this can really just encapsulate a message and be delivered to people to deliver that message. So mRNA, for this audience, I probably don't really need to go through this review, but just as a reminder, mRNA, and this is a part of the central dogma of biology, where you have DNA that has all your hereditary information, but when you want to use it, you then can transfer it into a message or messenger RNA, which can then be translated over to proteins. So mRNA, of course, contains instructions for cells on how to build all of their proteins or tools. Proteins then have a role in regulating all the functions of your cells and generally of your body. So when we think about mRNA vaccines in the context of cancer, what kind of message or mRNA would we want to deliver? And I think this is still something that's in the development phases, and hopefully we can hear a little bit more at the conference. But we can deliver mRNA that will help the immune system to find cancer, so maybe some targets, to create immune-related proteins, maybe to boost the ability of these cells to perform their functions, and lots of other different things that can be engineered. And we'll talk about one application in the following slides. So there's some applications to this mRNA lipid nanoparticle design. So you can have a very high production yield with this type of design. It lends itself to that capacity. It's also highly customizable. It's very easy to make these mRNA and can even be made on a patient-by-patient basis in some cases. Disadvantage to this type of design is mRNA is not broadly stable for a long period of time, and the protein product as well might not last very long in the body. So maybe you've delivered your message, you've gotten in what you need, but it's not necessarily going to last and have that impact for a long time, unless you've used it more like a vaccine in order to prompt your immune system to generate memory. So in that regard, there is some talks at the conference that have to do with this idea of neoantigens. So these are kind of what they sound like, neo meaning new antigens. So neoantigens or newly formed antigens don't exist in the body in general, but are generated in tumor cells. Remember that tumor cells have mutations. It's one of the hallmarks of cancer. And these mutations in the DNA can then get into the RNA and get into the final protein product. Even if they're not impacting the function of that protein, you might have a lot of errors that exist. And these amino acid sequences, these can actually be processed and shown to T cells in the context of antigens. So you might get a lot of antigens that are normal that the T cells recognize. They're like, oh, that's part of me. I don't need to react to that. But sometimes you can get development of these neoantigens that a T cell will recognize and be like, wait a minute, that's new. That's not part of me. I should react against it. And you can then get reactiveness to cancer in that way by these neoantigens that are not normally expressed in the body. So there's been efforts to develop neoantigen-based mRNA vaccines. So instead of the target that was used in the COVID vaccine, which was spike, you can actually use a patient-derived neoantigen. So there'll be a talks at the conference looking at how can we screen patients to discover what neoantigens they might have to then use that for vaccines. So a really broad concept that could lead to very, very interesting therapies. So you can have the T cells then respond to a neoantigen, and hopefully when they're educated, they can then kill these tumor cells expressing the neoantigens. Remember that when you have vaccination, this can lead to a better response when you see that again. So if we show a patient's T cells these neoantigens, maybe they can have a faster and higher response if they see them again. There is even a Canadian company that's been working on some of these mRNA-based vaccines. This includes this one, Providence, where they have been working on precision antigens for a variety of different tumors, as well as a variety of mRNA vaccines. So besides neoantigens, we can also think about in vivo engineering, which I think is the last plenary at the conference. And instead of taking T cells out of a patient, growing and expanding them in a dish, activating them, giving them the receptor, and then reinfusing them, what if we skip this whole thing? What if we actually just use the patient as the incubator themselves, making them into their own little factory? So this is kind of combining some of the concepts that we've talked about before. We can use different vectors or different packages in order to be able to deliver messages to the cells. So we talked about lipid nanoparticles, but those have some issues, as in you might not have that response for a long time. What if instead we used viruses in order to deliver messages? So there's some viruses that actually integrate the DNA that they're carrying into the host genome. So they have that message with them forever after that. So what if we actually give them instructions to make a CAR T cell receptor? We don't need to give that to the T cell out of the body. What if we just do it in the body? So you put that CAR DNA, those instructions, into the T cell, It's now in their genome, and then you have a T cell that is capable of making the CAR construct in vivo. So I've reflected this here just in words as well. A virus engineered to carry the DNA instruction to make a CAR receptor can enter a T cell, and that would be specifically targeted towards the T cells. The CAR receptor DNA can integrate with the normal T cell DNA, and the T cell can now have the information to make itself a CAR T cell. So there's definitely a lot of different in vivo approaches. I've taken a very general understanding of this, but definitely a little bit of a preview to what you might see at the conference. So we've covered a lot of information today, and I hope that it's been useful as a primer first to immunology, and hopefully have a little bit more information about the innate versus the adaptive immune system, types of immunotherapy that exist, including monoclonal antibodies and the important subcategory of immune checkpoint blockade, adoptive cell therapies, CAR T cells, TCR T cells, and TILs, as well as oncolytic viruses. And finally, we talked about a couple hot topics in science and immunotherapy, such as mRNA vaccines and neoantigens, as well as in vivo engineering. I'd be happy to take the rest of the time to answer any questions that you might have. You can also reach out to me by email or see me at the conference next week. Thank you for listening, and I'm happy to pause here to answer any questions.
Before we jump into Q&A, just wanted to say a quick thank you so much, Megan. That was a university quality lecture that you just delivered, and so fantastic and very comprehensive, and even for myself, a good primer heading into Summit. So thank you so much. And yeah, lots of celebrations there for that talk. So yeah, please put your questions in the Q&A or feel free to come off mute. We do have some time to address questions.
I think I did see a hand. I don't know if you're typing in the chat or if you want to unmute. So one question here, what are some of the biggest barriers to advancements of these new categories of therapies in a Canadian context? That's a big question. I'll admit to not being an expert on anything in the context of regulation. I have actually been lucky to have some experience with understanding clinical trials. Money is sometimes a big barrier. It costs so much money to run a clinical trial you wouldn't even believe. I didn't believe. So that can be an issue as well. Sometimes it's where the manufacturing is done. So I know in the context of CAR T cell therapy, for the most part, a lot of the manufacturing is done in the US, and they don't really have a ton of capacity to be able to manufacture for Canadians as well. So they have, I think, a limited number actually, I think it's both CAR T cells and TIL therapy that they can manufacture for Canada. But just getting some of that infrastructure up and running here in order to be able to do it. I know that BioCanRx actually supports a lot of that work here, and I promise they're not paying me to say that. It is very true. But they definitely need the money, need the infrastructure in order to get it running, for sure. Have we had other successful mRNA vaccines besides the ones used for COVID? Very interesting. Yes, I will admit to not really knowing the targets for them right now. There is actually the company that I talked about, Providence. They do have mRNA vaccines that are up and running for cancer. They have actually a beautiful story on there if you look at their website of a successful case of a patient that was treated with an mRNA vaccine for cancer. Very nice video there. mRNA vaccines actually were, I think, very much thought of in the context of cancer, and a lot of optimization that was done there before COVID-19 had emerged as well. So some of that was a little bit of crosstalk between vaccination for viruses as well as cancer immunotherapy. I'm sure there's tons of our other targets, not ones that I know of offhand, though.
Megan, if I can maybe just jump in with a question, although I think another one has come in, but you mentioned, so your research is about metabolism and its connection to the effectiveness of these therapies. And given that these are living drugs, can you speak a little bit to the role of metabolism?
I absolutely can speak to the role of metabolism. I'm always happy to. We think about metabolism as fuel. So you think about you put gasoline in your car, you want to put the right type of gasoline to make it go properly. So every cell in your body is going to need fuel in order to run itself. And T cells, they have so many functions. So they need the right fuel in order to be primed. And the tumor microenvironment is so challenging for immune cells like T cells in order to have their functions. So really, there's a lot of nutrients that are being used by tumor cells, being used by others in the environment. You have hypoxia going on. You have lots of waste byproducts as well. So you really want to kind of examine how we can best fuel the T cells to survive in this environment and to be able to perform their function. So we are kind of trying to link that. Let's give them the right fuel to survive the journey, and how can we use that fuel in order to let them perform their function? I think that hopefully answers the question.
Yeah, awesome. Thank you.
Yeah. Okay, the question that's come in from Meg here maybe is one for BioCanRx about plans to make this more accessible and moving knowledge to publics, journalists, and politicians. I don't know if you have an answer for this now.
Oh yeah, I guess am I on the spot here? Do you mean specifically, I mean the webinar, absolutely, but yes, part of our mandate is to communicate the value of these immunotherapies to the public, and we have a lot of channels that we use to do that. So one, we have the Cancer Community Partnership, where we have a group of cancer advocacy groups, and working with them always to communicate about the latest research and the challenges that someone already brought up about advancing these to the clinic. And then also communicating to government. And yesterday, our president and CEO was in front of government, really advocating for the importance of this work and translational research in Canada. So if you want to join that aspect, and get in touch, and we are always looking for more voices to be part of that.
Another question, what do we know about metabolic fuel tumors rather than for fueling T cells? Absolutely, definitely an ongoing area of research. We sort of collaborate with a lab that's looking at the tumor side of metabolism. Kind of funny, T cells and tumor cells are actually very similar in terms of how they do their metabolism. So a lot of the research that's being done for one is actually applied to the other as well. There's one concept people talk about, aerobic glycolysis. This is probably more of a biochemistry talk, so I'll be very brief. But usually when you have oxygen, you go through glycolysis and then you go into the TCA cycle and the electron transport chain. I'm so sorry if I'm giving you guys trauma flashbacks to your biochemistry class. But in that context, you need oxygen. And usually that's what you're doing to get a lot of ATP generated. In the context of really wanting to pump the machine very quickly, sometimes they'll kind of not use the end part and just use glycolysis in order to be able to fuel things faster and to use other kind of offshoot pathways to get intermediates. So there's a lot to know and a lot to learn, I think, about both metabolism for tumor cells and metabolism for T cells. And the resources that might be available to these cells, what's coming in, how are they managing hypoxia, what are the nutrients that are kind of limiting in that environment. So definitely tons to know. Question also from Con. The last name.
Sorry, I didn't realize that my first name wasn't spelled out all the way.
That's okay.
I guess my question to you is, what keeps you up at night about all of the work that you do and knowing what you know?
Yeah, that's definitely an interesting question. I think many things. Definitely the realities of science, and I'm sure many people in the call share this, is just getting things work in science can be very difficult. But also, like, I have one project that's really dedicated to understanding T cells at a more fundamental level. It's a lot more mouse work. And I have one that's really geared towards and using patient samples. If you find me at the conference at my poster, I'll actually be talking about that second project. But really, that project and like, how do we get this towards patients? And I think coming from a grad student perspective, I don't always know, and I've been very fortunate to work with a clinician scientist, to work with BioCanRx, to talk to patients and to understand some of their perspective. But it's like, you know, I'm head down in a petri dish in the lab and I'm looking at my cells, and how can I really make sure that the impact that I'm having is going beyond those cells in a dish, and I'm really keeping in mind sort of the implications of the work that we're doing, and allowing this to really be a very robust scientifically, but also to have impact beyond that. So maybe not keeping me up every day, but I do come back to that question as I put some of my work together. Any last questions?
Okay, well, if there are no further questions, Meghan has already invited you to send an email if one pops up later, or if you're at Summit for CI, drop by your poster, come talk. But thanks everyone for joining us today. Again, Meghan, this was a fantastic talk, and I think so informative for everyone who's attending. So thank you so much for putting this together and explaining it so clearly. I think that's so helpful especially for immunology, which can be quite complex. So thank you for doing this. I'm going to just put up a quick QR code. We do have a post-webinar feedback survey. So if you take a minute or two just to share your feedback, we're always interested in hearing from you of what else you want to learn about, and whether we can provide that information. So please share your feedback. And if you want to keep in touch with BioCanRx, follow us on social media, join our newsletter. You can hear more about the research that's going on and the stories that come out of that. So thank you everyone for attending, and hope to see you at Summit if you'll be there in person.